This shared resource has been under the direction of Dr. Michael Verneris since 2012. Molecular and Cellular Therapeutics is a CGMP-compliant production facility that provides development and manufacturing of cell and tissue-based products, monoclonal antibodies, targeted toxins, and other recombinant therapeutic proteins. MCT performs cell purification and large-scale cell culture using bags, cell factories, bioreactors, and other specialized equipment. The Translational Therapy Laboratory is a central laboratory that receives and processes research blood samples from patients enrolled in clinical trials. It also provides immune monitoring services and biorepository management. The biorepository function of TTL includes the storage of viable cells, DNA, RNA, and serum or plasma collected from patients enrolled in clinical trials.
In recovery after transplantation. And typically, one of the types of transplants is in a double umbilical cord blood transplantation. Even though this can be very effective treatment, one of the most common problems is still relapse. Our lab also studies natural killer cells, and this is a type of cell that can kill cancer cells. We know that this is one of the cells that comes back very early after transplantation, and so we thought that this could be involved in the transplant recovery. We know that it's important in recovery after transplant, and so we thought maybe this is also due to related to the proliferation of the NK cells and how fast they grow after the transplant. So we have a resource at the University of Minnesota for the Translational Therapy Lab where they actually have a repository of all the different patients, or many of the patients that are on clinical trials undergoing different types of transplant. So the great thing is that we could decide we want all the patients that have double umbilical cord blood transplantation at what time points that have had what diseases, and ask for this data. And so in addition to having those samples, not only serum samples but also cells, the Translational Lab typically does an initial analysis when they get the samples from the clinic. So they have done a preliminary analysis of the proliferation using different markers, and so we can use this initial data to inform our decisions.
I chose this project because this is a large study, probably the largest study that this resource supports. Over 1,000 patients have been enrolled on this study, and blood is drawn from those patients at about 8 time points during the course of their bone marrow transplant. And so this study has produced at least 8,000 different data points for which we have blood and serum and clinical information, which has all been merged together. So I think it's a great example of how this resource provides really high-end support to a complicated clinical ancillary study. The main way that the core interfaced with this particular researcher was, number one, to do analysis of the samples that were provided from the clinic, and the analysis was really mostly flow-based, so establishing the panels and providing that data. As well, the core also collected clinical samples and froze those down, both peripheral blood mononuclear cells and serum, which was then provided back to the researcher when they asked for it for their kind of bulk assays. This work couldn't have been done without this core. In other words, having a single investigator try and track down blood samples at various times in the clinical course of a patient undergoing bone marrow transplant, and getting those samples and storing them and analyzing them simply couldn't be done by one investigator. It really does need to be a concerted core-based effort to do this. And so without this core, this research could not have been done. I'm quite convinced of it since I've tried to do it myself in the past, hence the creation of this core.
To work with the Translational lab and find out what their protocol was, and they worked with me to replicate that protocol in our own research so we could be as consistent as possible. And what we found is that in some cases we could use the same reagents, in some cases we were recommended to use different reagents. So it was great because we had a very organic process of finding out the best protocol to perform our experiments. In addition, we were able to troubleshoot with them and look at our data side by side to see where we could make these changes or optimizations.